In this section, we are going to be dealing with the foot tests, identifying the various molecules in various food samples. In this presentation, we will briefly introduce ourselves into carbohydrates and then take the first test, which is the test for reducing sugars. My name is Lenya Jude Fanba. I am a holder of a Bachelor of Education degree in Life and Earth Sciences from the University of Marua, Cameroon, and a Doctor of Philosophy degree in Clinical Biochemistry from the University of Chang, Cameroon. To better understand the tests and experiments under carbohydrates, let us first briefly look at some general knowledge on these molecules. Carbohydrates are organic compounds made up of carbon, hydrogen, and oxygen. They have a high oxygen content with the ratio of hydrogen to oxygen, like that of water. Carbohydrates primarily serve as energy sources and structural components in living organisms. Carbohydrates can be vaguely classified as sugars, polysaccharides, complex carbohydrates. Sugars are simple carbohydrates. They are small, soluble, sweet, and crystalline molecules. They are divided into two groups, namely monosaccharides and disaccharides. Monosaccharides, single unit carbohydrates. They are carbonyl compounds with the aldehyde or ketone groups. Monosaccharides with the aldehyde group are called aldoses, while those with a ketone group called ketones. The alpha carbon, that is, the carbon atom holding the functional group, is thus C1 in aldoses and C2 in ketoses. Common monosaccharides are trioses with 3 carbon atoms, for example, glyceraldehyde and dihydroxyacetone seen in glycolysis. Pentoses with 5 carbon atoms, for example, ribulose the carbon dioxide acceptor in C3 photosynthesis, ribose and deoxyribose in RNA and DNA, and hexoses with 6 carbon atoms, for example, glucose, galactose, and lactose. Recall that in solution, pentoses and hexoses usually exist as rings. The alpha carbons are indicated here, and due to rearrangement of the atoms, it has a hydroxyl group which helps in defining some characteristics of these molecules, for example, the alpha and beta isomers. Disaccharides are sugars formed from a condensation reaction between 2 hexose monosaccharides to form a glycosidic bond. Examples are maltose, the sugar in partially germinated or malted cereals formed from 2 glucose molecules, lactose, the sugar in milk formed from a glucose in a galactose molecule, and sucrose, which is table sugar, and also the sugar in sugarcane formed from a glucose and a fructose molecule. Sugars can be classified as reducing and non-reducing sugars. Reducing sugars have free aldehyde or ketone groups that reduce Benedict's or Fehling solution. That is, the hydroxyl group on the alpha carbon is free. All monosaccharides and some disaccharides, for example, lactose and maltose, are reducing sugars. Non-reducing sugars do not have free aldehyde or ketone groups and thus do not reduce Benedict's solution. Sucrose is a common example because the hydroxyl groups in the alpha carbons of both glucose and fructose are used to form the glycosidic bond that links them. Polysaccharides are polymers of monosaccharides, especially glucose. Storage polysaccharides serve as energy reserves, for example. Starch is the storage carbohydrate in plants made up of alpha glucose. There are 2 forms, amylose, which is linear, and amylopectin, which is branched. In animals, the storage carbohydrate is glycogen, which is highly branched for rapid energy release. As for structural polysaccharides, they provide support and protection. For example, cellulose in plant cell walls, composed of beta-glucose units, giving it high tensile strength. Complex carbohydrates, on the other hand, contain additional elements like nitrogen, sulfur, or phosphate. Examples include chitin, a nitrogen-containing polysaccharide found in insect exoskeletons and fungal cell walls. Peptidoglycan, a structural carbohydrate in bacterial cell walls that contains nitrogen. Glycoproteins and glycolipids, carbohydrates attached to proteins and lipids in the cell membrane, playing key roles in cell communication and immune responses. Let us now look at the test for reducing sugars. The test is based on the fact that reducing sugars have free aldehyde or ketone groups that can donate electrons. When heated with Benedict's or Fehling solution, these sugars reduce blue copper 2 ions to insoluble red or orange copper oxide precipitate. This test is done using either Benedict's or Fehling solution. Benedict's solution is an alkaline solution of copper 2 sulfate, sodium carbonate, and sodium citrate. Copper 2 sulfate provides blue copper ions, which get reduced to red copper, 1 oxide in the presence of reducing sugars. Sodium carbonate maintains an alkaline pH, preventing copper 2 ions from precipitating as copper hydroxide. Sodium citrate forms a complex with copper 2 ions, keeping them in solution before reduction occurs. As for Fehling's reagent, they are 2 solutions. Fehling's solution A contains copper 2 sulfate, which supplies copper 2 ions. Fehling's solution B contains sodium potassium tartrate and sodium hydroxide. Sodium potassium tartrate acts as a chelating agent, preventing copper 2 ions from forming insoluble copper hydroxide. Sodium hydroxide provides an alkaline medium for the redox reaction. We are going to use a set of materials and solutions, including a food sample, for example, germinating seeds, sugarcane milk, a solution of a sugar, etc. A mortar and pestle will be used to crush the food if necessary. A filter paper and beaker will be used to filter the extract. The extract can also be obtained by decantation. Syringes to measure volumes of solutions. A test tube to hold the mixture. A test tube holder to hold the test tube during heating. A boiling water bath for heating the mixture. A pot and a stove can also serve the purpose. A wall clock with a sweeping second hand for timing. As for the solutions we need are going to use distilled water to prepare the extract. We also need Fehling's solution A and Fehling's solution B. Benedict's solution can be used instead. You may be required to make an extract from a food sample before testing it for reducing sugars. Take a small quantity of the food, say 10 gram, or a 5 centimeter length of peeled sugarcane stem, or still 10 germinating maize seeds, etc. Crush the food in a mortar and pestle. Add 10 to 15 milliliters of water depending on the quantity of food substance crushed. Mix and filter out the extract using a filter paper into a beaker. Measure 2 milliliters of the solution into a test tube using a syringe. Add 1 milliliter each of Fehling's solutions, A and B, to the test tube using a syringe. Add 1 milliliter each of Fehling's solutions, A and B, to the test tube using a syringe. Mix the solutions gently and place the test tube in a boiling water bath for 3 minutes while shaking from time to time. Sample report of this procedure would be: if you started with a food sample, you will have to describe the process of getting the extract as follows. 10 germinating seeds were crushed using a mortar and pestle. 10 milliliters of distilled water were added to the crushed grains. An extract of the crushed grains was filtered into a beaker using a filter paper. If you started with provided solution, say solution X, start reporting from here. 2 milliliters of the extract were added to a test tube using a syringe. Or 2 milliliters of solution X were added to a test tube using a syringe. If you started with a solution, 1 milliliter. Each of Fehling's solutions, A and B, were added to the test tube using a syringe. The test tube was shaken gently to mix the solutions. The mixture was placed in boiling water bath for 3 minutes while shaking from time to time. As results, we have: for a positive test, upon heating, the color of the mixture changed from blue to green to yellow to orange and finally to brick red. For a negative test, upon heating, no color change occurred. Note that if the solution is too concentrated, the brick red precipitate is instantly formed with no apparent color transitions. Also, a less concentrated solutions will give green to orange color and not brick red. For conclusion, if there was a positive reaction, we say the extract or solution X contains reducing sugars. If there was a negative test, we say the solution or extract does not contain reducing sugars. We have come to the end of this presentation. Our next presentation will be to evaluate the reducing effects of diluted solutions of a reducing sugar, for example, a glucose solution. Thanks for your time. Hope you enjoyed following up the presentation. Please subscribe to this channel, like, and share. By subscribing, you will be getting notifications when new presentations are available.